Good day. This is the Professor, CBMD PhD, and today's Monday, May 8th, 2023. It is 5:00 and 17 seconds, AM Eastern Daylight Time. And today's research, Glioblastoma. So let's hop to it here. As this gel stops brain tumors in mice, could it offer hope for humans? Date April 24th, 2023. Source: Johns Hopkins University. Medication delivered by a novel gel cured 100% of mice with an aggressive brain cancer, a striking result that offers new hope for patients diagnosed with glioblastoma, one of the deadliest and most common brain tumors in humans. Despite recent technological advancements, there is a dire need for new treatment strategies. They think this hydrogel will be the future and will supplement current treatments for brain cancer. The research team combined an anti-cancer drug and an antibody in a solution that self-assembles into a gel to fill the tiny grooves left after a brain tumor is surgically removed. The gel can reach areas that surgery might miss, and current drugs struggle to reach to kill lingering cancer cells and suppress tumor growth. The gel also seems to trigger an immune response that a mouse's body struggles to activate on its own when fighting glioblastoma. When the researchers rechallenge surviving mice with a new glioblastoma tumor, their immune systems alone beat the cancer without additional medication. The gel appears to not only fend off cancer, but help rewire the immune system to discourage recurrence with immunological memory. And still, surgery is essential for this approach. Applying the gel directly in the brain without surgical removal of the tumor resulted in a 50% survival rate. The surgery likely alleviates some of the pressure, allows more time for the gel to activate the immune system to fight the cancer cells. The gel solution consists of a nano-sized filaments made with paclitaxel, an FDA-approved drug for breast, lung, and other cancers. The filaments provide a vehicle to deliver an antibody called ACD47, and by blanketing the tumor cavity evenly, the gel releases medication steadily over several weeks, and its active ingredients remain close to the injection site. And by using that specific antibody, the team is trying to overcome one of the toughest hurdles in glioblastoma research. It targets macrophages, a type of cell that sometimes supports immunity, but other times protects cancer cells, allowing aggressive tumor growth. One of the go-to therapies for glioblastoma is a wafer, commercially known as Glidel. It is FDA-approved, biodegradable polymer that also delivers medication into the brain after surgical tumor removal. As Glidel showed significant survival rates in laboratory experiments, but the results achieved with the new gel are some of the most impressive the researchers see. They don't usually see 100% survival in mouse models of this disease, thinking that there is potential for this new hydrogel combination to change that survival curve for glioblastoma patients is very exciting. The new gel offers hope for future glioblastoma treatment because it integrates anti-cancer drugs and antibodies, a combination of therapies researchers say is difficult to administer simultaneously because of the molecular composition of the ingredients. And this hydrogel combines both chemotherapy and immunotherapy intracranially. The gel is implanted at the time of tumor resection, which makes it work really well. In addition to other brain tumor therapies currently in clinical trials, emphasize the challenge of translating the gel's results in the laboratory into therapies with substantial clinical impacts. The scientific investigators and scientific researchers say the challenge now is to transfer an exciting laboratory phenomenon to clinical trials. And this is the professor, Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, Have a great day.